cloning is where you make an exact copy of the DNA. And molecular cloning is where you can make an exact copy of a protein. And so a good example of molecular cloning is where you take a human gene and you insert it into a bacteria. Bacteria are known for easily taking up the genetic material from their environment. And it's pretty easy to insert a gene into the bacteria, and the bacterial DNA is not chromosomes, but it's circular plasmids, they call it. But once the human gene is in the bacteria, then the bacteria reproduce, and they reproduce asexually. So they reproduce asexually, and they produce human protein. So you can actually use bacteria as a factory for producing human protein. And this has been done for a variety of things, but one thing that it's famous for is the production of insulin. So insulin is a hormone that is needed by people that have diabetes, specifically if they have juvenile or type 1 diabetes. Their pancreas cannot produce it. And so instead of getting it from pig pancreases, right, we can use the gene, put it into a microbe, and have the microbe produce the insulin for us. Okay. So that is an example of molecular cloning. And in your book, there's a diagram of this. So this is my foreign DNA. This would be my human, right? We put the gene into, so this would be my human gene, into the circular DNA of the bacteria. We allow the bacteria to reproduce, right? And then we figure out which ones have taken up the DNA, and then we let them reproduce, and then they will eventually produce human protein. And that's a way that we can clone human protein, is through the use of bacteria. Now, we're going to talk about therapeutic and reproductive cloning. These are types of cloning that are both have the same basic premise for how it is done, but the result, what we're trying to get out of it, is dramatically different. Okay. So in therapeutic cloning, you would take a somatic cell and you remove the nucleus. So that's the genetic material I want to clone. Somatic cell would be a body cell from me. So if I want to get stem cells, this might be how I would do it. So I take my somatic cell, I remove the nucleus, and I put the nucleus into an egg that has had its nucleus removed. Okay. It's important that I have an egg because I need a big cell, right? I need a cell that is going to be able to divide really rapidly. And so then I stimulate it to undergo mitosis. And generally it's like an electric shock they give it. And then I get a multicellular embryo. Now, that embryo contains stem cells, and stem cells are unspecialized cells. So these are unspecialized, and they can be used to treat disease. Right? So I could use those stem cells, and you can put them back into the person. Okay, so stem cells are used therapeutically. To treat disease. So for example, maybe if I have Alzheimer's and my nerve cells start to die, maybe I could take a cell, produce a multicellular embryo that is identical to me, right? Those stem cells could then be put back into my brain, and the stem cells would divide and they would rebuild my nervous system. Okay? So that is how it is used therapeutically. Okay? Now, otherwise, this multicellular embryo, right, could be used to produce another individual. So if I wanted to clone myself, I could take that multicellular embryo and I could put it into a surrogate. So I could put it into a female's uterus, and then she would grow another me, right? That has not been done. It's not been shown that it has been done in humans, but it has been done in other organisms. And does anybody know what was the type of organism where reproductive cloning was first? Yes, sheep. Okay. So this is what we call reproductive cloning. Okay. And the first one was a sheep named Dolly. Oops. Okay. So why was the resulting sheep, clone sheep, called Dolly? Anybody know? It's very silly. Dolly Parton. And she had big breasts. Dolly Parton has big breasts, right? Is she still alive? I'm not sure.
Yes.
She's still alive. So she has big breasts, right? So what they did is they took mammary gland cells. These are specialized, but they took the nucleus out of it and they put it into an egg cell. Okay? Notice that they used two different breeds of sheep. And the reason why they did that is they wanted to make sure that the resulting organism was truly a clone. So if it came out as a Finn Dorset sheep, then they know it's a clone. If it came out as a Scottish blackface, then they know that they didn't do it right, right? There was another embryo present or sitting in the uterus, okay? So they give it a little pulse, right? And you start to develop an embryo, and they put it into the surrogate ewe, and then the resulting sheep was Dolly. One of the interesting things about Dolly is that she died early. She seemed to age faster. So why do you think she might have aged faster? Yeah.
Is it because of Shine does have the genome?
She has the whole genome. Yeah. It was an older cell. It was an older cell, and what's about the older cell? Mitotic clock, right? So she only lived to be 6 instead of 10 or 11 or 12, which is typical of sheep. And so she had a shortened mitotic clock because her genetic material was taken from an adult, right? And so that's a problem, is if you take the genetic material from, like, you want to clone an old person and you take their genetic material, where their mitotic clock is in the embryo when you can be wild, right? So that's a problem with cloning old people, is that they're unless maybe you can figure out how to add telomerase and get the telomeres to expand back up. Okay.
So what significant findings did they find in Dolly besides the mitotic clock?
Well, they found that the mitotic clock was a big one, but her offspring are perfectly normal. So she reset the mitotic clock in the cells that gave rise to her eggs, like we all do.
Like, what's it called? I'm sorry, when a woman becomes infertile, like she was cloned post infertility, would Dolly be infertile as well? Because
the one thing that you might think of is, I just realized this, is the imprinting. the imprinting, actually the imprinting would be the same. It would be normal imprinting because the mom had half the chromosomes imprinted as the father and the female, and so the imprinting would be normal. Do you understand that? It's normal imprinting because it was from an adult cell that didn't give rise to her sperm. I don't know, but they know her progeny have been healthy, and so they know that it didn't cause any long-lasting problems. It was just in Dolly herself.
Her pregnancy was normal too.
Her pregnancies were normal. Okay, so there are a couple of things that I need to record. So it should be a short, maybe 20-minute lecture. And so I will get that up and on the Canvas website. So all we need to talk about really is transgenic organisms, genetic engineering, and gene therapy. That's what we're going to talk about. So everybody has a copy of the review sheets. Nobody came in late? Copy of the review sheet. Did you get a copy? Okay. This is for the final. Homework? Okay. I will take that.
If he can get a sheet, he gets fun because he's passed all the